Can you? Okay. Are you able to see the screen? Are you able to see my screen? Hello.
Yes, sir, it is visible.
Okay. So, yeah. So, as the terminology goes, CRISPR is nothing but it is a clustered, regularly interspaced, short palindromic repeats of gene sequences, and these gene sequences have specific properties of acting at particular sites of in a DNA sequence. And moving forward, the evolution, a little bit on the introduction on the evolution of about CRISPR. It was discovered in 2012, and finally led, as any other scientific discoveries, it led to a Nobel Prize awarding. And thereafter, its protein variants have been discovered. The initial variant was CRISPR-Cas9, the enzyme, and then 12 and 13 also were identified. Thus, in applications where 12 and 13 are used, there is no editing required. It can be used directly. Advantages is it is high specific speed, low cost versus PCR. It is adaptive immune system for bacteria. It was repurposed for genome editing and detection. Bacteria in general have this kind of adapted this kind of immune system, which is in short called as the CRISPR. So whenever there is a attack or any kind of foreign body attack from the, for example, any bacteriophage or any kind of infection to the bacterial cell, it is ready with this kind of immune system, which is called as CRISPR. Now, the key components that are used are engaged during the CRISPR action is the Cas9 enzyme, which is the molecular seizures. And there is a specific RNA called as the guide RNA, single gene RNA. So, and that targets the specific DNA. The mechanism is the guided RNA. What does it do is it combines with Cas9 complex and binds and cleaves the DNA at precise locations. Okay. Madhvi, can you put this? Yeah. Okay. Sorry. So it cleaves the DNA at precise locations via the protospacer. Protospacer is nothing but, again, it's a short sequence of nucleotides, which is available on which is required for the action to be completed. And this protospacer is adjacent to, it's an adjacent motif present to the sequence for intended to be acted upon. Is my slide moving?
Yes, sir.
So, to briefly, I took a piece of a single small YouTube video that's already most of you might have seen. But for those who are not aware of what is CRISPR, this will probably explain you what it is. Let's have with us a short video, and I'm sharing this with you.
ഇന്നത്തെ പ്രത്യേകം എന്താണെന്നറിയാമോ? കൊതി ഉണർത്തുന്ന മണം.
ഇപ്പൊ നമ്മുടെ ഫാമിലിയുടെ ഫേവറേറ്റ് മാഗി, കേരളാസ് നമ്പർ 1 നൂഡിൽസ്. മാഗി കേരളാസ് നമ്പർ 1 നൂഡിൽസ്.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our
So have you actually read what is in a document? If we suspect we have misspelled a word, suppose if we are typing a Word document and we have a doubt that the spelling could be wrong, what we do the find and search version, and in the find version, what we do is in this find version, we type the word and we can look for the correct version, right?
Within our DNA, that function is taken on by a system called CRISPR-Cas9.
So in the DNA sequence, if just such kind of errors, spelling mistakes happen, so in bacterial DNA, what happens is this kind of CRISPR-Cas9 immune system is inherently inbuilt in the bacterial system. But now we are using this technology or this kind of system to eliminate diseases, to mutate sequences, to invent new sequences. This kind of technology is being used through a technology called as CRISPR-Cas9.
9. CRISPR is short for clustered, regularly interspaced short palindromic repeats.
So these sequences, these short sequences, these are clustered. These are regularly clustered in the sense that nucleotides are in small clusters. It could be 2 to 7, 6 to 5 to 8. In short palindrome repeats, it will be there. The cluster is this unit of sequences, and they are irregularly interspaced. So if a particular side intervals and along the sequence, gene sequence, they are available. And they are palindromic. I'm sure you people know the meaning of palindromic. That is, from either side you read whether it is 5' to 3' or 3' to 5', the sequence remains the same.
CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA.
So here, I hope you people have understood why it is using this kind of complement the RNA sequence against the DNA sequence. So that is, in other words, which is called as the cDNA.
The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care, or even cure lifelong inherited diseases.
Imagine conducting every part.
So moving on. Is my screen visible? Advi, is my screen visible?
Yes, sir. It is visible.
Okay. So CRISPR technology derived from bacterial immune systems. It involves precise DNA editing using guide RNAs and Cas enzymes. This package's requirement is a pre-requirement. You need to have a guide RNA against which the DNA needs to be anchored and removed or cut. And the catalytic role played by the Cas enzyme protein is significant here. And CRISPR screening is high throughput method to test thousands of genes functions via pooled guides. Once when you have a lot of CRISPR technology used immune systems or sequences to study, then once you created a lot of CRISPR gene database, then you need to have a screening method. So that can be done. That can be done easily by pooling the samples that you have studied of interest. As I mentioned earlier, the CRISPR-Cas9 mechanism in bacteria and the gene editing. These are the components. There will be a DNA sequence. There will be a DNA sequence, and there will be a CRISPR RNA to which the PAM will attach, and then the cutting will take place, in which the transcribed the trace RNA and the Cas enzyme act together. So these are the important components that are required to complete the CRISPR-Cas9 ecosystems. We need to have the PAM sequence. There is an enzyme, there is a guide RNA, and there is a CRISPR-Cas9 editing that happens. So the core components are CRISPR, which I said earlier. It's a gene sequence which repeats over the sequence of the bacteria or the piece of DNA sequence which we are interested to study. Cas9, it's an enzyme which is required to cut the DNA at specific sites guided by the RNA. Then we have the CRISPR RNA. It's a small RNA derived from the CRISPR array that matches target viral DNA sequences. Then precursor CRISPR RNA is required, which lays the template for cutting of the sequence. Then there's a tsRNA, transcript activating CRISPR RNA. It pairs with the CRISPR RNA to form a functional guide for Cas9. So here you can see the gene is there, gene of interest is there. The action points are where the Cas9 and the PAM access points takes place. This is the precursor RNA. This is the first sequence, first line of sequence, which we need to have the act upon the RNA. Then we have the transcript activating CRISPR RNA. There are sequences where the point to be noted is where there is a Cas operon. Wherever there is an operon, which is area of operations will depend on the operonic area where the Cas enzyme can actively act upon the array. Moving on, the guide and targeting terms, which is generally used in CRISPR technology, is the single guide RNA, which I'm sure you people might be knowing. It's nothing but a synthetic fusion of cRNA and tracer RNA for simpler targeting in lab applications. Then the protospacer, which I mentioned earlier, PAM, it's another sequence, which is, for example, in NGG, it is for Cas9, required next to the target for Cas binding and cleavage. The protospacer I mentioned earlier, it's again a DNA sequence in which the virus or genome that matches a CRISPR spacer. So these are the variants and techniques in which the CRISPR formats of action lies in different kinds of, suppose CRISPR-R, activation uses modified Cas to upregulate gene expression. CRISPR-RI is an interface CRISPR. It uses the dCas9. dCas9 is the deactivated Cas9 or the death Cas9. It is actually a Cas9 enzyme which is deactivated to repress gene expression without cutting DNA. The dead Cas9 is a nuclease inactive version for regulation or imaging and not cutting. Then there is Cas12 or CPF1. This is alternative Cas enzyme that creates staggered cuts and processes its own CRISPR RNA. The editing processes happen through three routes, which is called as NHEJ, which is non-homologous end joining, and then error-prone DNA repair pathway causing insertions, deletions for gene knockouts. And there is HDR, which is homology-directed repair. It is precisely happening. It functions in a precise manner, with precise repair using a donor template for gene insertion or correction. So, and then there is the ribonucleoprotein, which is a pre-assembled complex of Cas protein and guide RNA for delivery. So, photospacers role in CRISPR Cas9 targeting. This is the PAM sequence, and then these are the areas or functions that is required to happen. The binding signal for Cas nucleases, then it is required for initial binding, and then it distinguishes from self and non-self. If it's a self DNA or a non-self DNA. Then if it is a self DNA, it's prevent cutting from host genome. Then required for binding, initial binding, unwinding of DNA target. This is the workflow that has to happen when there is a CRISPR-Cas9 activity happening. Targeting and immunity, then PAM dependency. These are all factors in which a CRISPR and Cas9 environment functions. Then source of spacers, difference from spacer, PAM essentiality. Without PAM, Cas9 will not bind or cut the photospacer. So this is an overview of a brief history of developing the main CRISPR-Cas9 editors. It's somewhere started around 2013. And in using various genome editors. And today, as on date, there is a huge amount of data sets available in public domains. Large amount of work has been done by the Chinese and Americans. most of these data sets are coming from these two countries, but these data sets are accessible globally, and a lot of research is happening. As from 2012 or 13 to 20, as on date, a lot of refinement has taken place. The initial non-clarity about using CRISPR now is slowly getting better, because CRISPR, as we all know, it is creating a lot of cost. The cost of improving designing a CRISPR-based gene or a technology is much beyond than any normal amplification technologies compared to RT-PCR or things like that. Another point that challenges the CRISPR environment is the instability of these molecules or the nucleotides when they are in a CRISPR environment. When I say CRISPR environment, this is not in a in a proper order of function that can be used by every microbe or every experimental technique that we intend to do so. These need to be designed and tailored as per the specific need of the preset sequence that we are aiming to work with. So a lot of time and resources are required while designing the initial sequence. So what happened is, over a period of time, instead of doing this R&D in the wet lab, now a lot of in silico work has happened, and in silico based testing has happened, and hence a lot of other CRISPR sequences are also available for research. Having said that, what happened is, if I get an in silico sequence using in silico studies or bioinformatics tools, and when I actually get to a lab to use that sequence or develop that sequence, there are difficulties in employing or using these sequences at the ground level when it comes to actually practical implications, whether it is cutting or binding or hybridization or things like that. So there is difficulties in handling the CRISPR sequences. So this is briefly a workflow for applying genome editing tools to modify genome sequences. So this is the first step, is to design a guided RNAs with high efficiency and specificity. These are the tools that are available, CRISPR, chop, and things like that. So these are tools that are available, and they need an input target gene, and they need to work on a in silico based designing structure. That is, I don't know how many of you have done the bioinformatics hands-on gene editing or gene designing things. People who have used BLAST and various types of BLAST or protein sequences, they understand what I'm trying to say. When you actually use the sequences or taken from the public databases available from GeneBank or PDB or things like that, and then try to redesign things, and then try to analyze the outcome of the new genes that is happening, and is it actually going to work in the real-time world or not. All those things, the practical feasibilities and the objectivities of functioning with these sequences in the wet lab is still a challenge even in recent times. Then the second step is delivering the CRISPR-Cas system into the cell. So once you have a well-screened and well-controlled sequence, this we need to, as I said earlier, we need to put it into a functional mode. So what we do is we get it into a plasmid and then try to grow the environment, and then after maturity of the plasmid and the sequence is completely available for screening, it is subjected to further screening and sequencing. Once that is done, then we evaluate the application, the utilities of the CRISPR-Cas9 genome for further editing or further requirements as per the experimental requirements. So these are some of the common applications of CRISPR technology. In molecular diagnostics, it is there. In gene therapy, it is there. In basic research, it is always there. It has been there, and it will continue to be there. Diagnostics and agriculture is also another area which is there. So if any of you are working in these, and if you have any particular problems or questions that we can discuss at the end of this talk, that will be great. Briefly, molecular diagnostics, gene therapy, basic research, diagnostics, agriculture. Gene therapy and molecular diagnostics is something which is catching up very fast, and even in diagnostics also, but both plant diagnostics and human diagnostics. Then we have so recent advancements. There have been some technological advancements in the CRISPR beyond Cas9. So in the initial protein that used to set well in the CRISPR sequence for easy binding and then forward cutting was Cas9. However, as I mentioned earlier, Cas12 and Cas13 also have come in, and further subsequent proteins are also evolving as research is moving. So the base and prime editing, which I think you all might be aware of. Then we have Cas variants, as I mentioned earlier, Cas12 and Cas13 are there. In that also sub variants are coming up, and as research is moving faster, we are evolving with new type of Cas sequences. Then epigenetic and activation tools are also available. Therapeutic progress is happening. Prime editing, as far as therapeutic progress is concerned, prime editing enters trials for rare diseases. In vivo editing reduces chemo needs for blood disorders. These also enable crop improvements and for safer CAR-T autoimmunity in terms of therapeutic progress. Now, there are, as I mentioned earlier, the ethical concerns with CRISPR gene editing. We also know that there are problems with if this technology has fallen into the wrong hands, it could be misused. The live examples of these kinds of activities were found during the US invasion of Iraq, when weapons of mass destruction were destroyed. At that point in time, it was technologies like these that new bioterrorism microbes were being developed and used against were intended to be used against humans, and these are the kind of pitfalls that such technologies have. So that's why one of the reasons when these kind of products go to the regulator scrutiny, whether it is the biotechnology regulators or the biopharma regulators, when these kind of products, whenever they come to know that there is a genetically modified crop or there is a genetically modified gene and which is going to be inserted into some microbe like Pichia or Pichia pastoris or any kind of E. coli or any microbes, they actually look at it very in detail, and through the lens of a regulator. If it doesn't pass, then that means there is some problematic or ill-intended use of these technologies. But having said that, in India, public focus on public health had taken place, and for TB there has been application of CRISPR, and in various IVDs. What happened is one of the TB diagnostics, single nucleotide polymorphism was detected. So rapid SNP detection for drugs resistance was done using the CATG gene in MDR TB, multi-drug resistant TB. So the CRISPR, the research is moving on and it is growing. And as on date, it stands at 88% sensitivity, the CRISPR TB detection kit, which is equivalent to 3.13 STFU per ml. Then, so as I said, tried to pick up some case studies that have been developed using CRISPR-Cas9. It was developed by CSR IGB. This is Tata has developed this COVID-19 detection kit, and it was one of the early diagnostic rapid test kits launched in India during the COVID-19. The technology is behind it is Feluda. It's a fluorescent assay. So, but the cost of duplicating this assay and multiplying this assay on a faster scale, on a larger scale, the cost factor came into picture. CRISPR at that point of time in technology and the sequence that they had in hand, both technology and manufacturing on that scale, which the pandemic struck us, was not possible. That's why the other kind of rapid assays took over. But this was one of the first initial products of CRISPR technology available in India, and particularly during COVID time. Although it was the first product, but as I said, commercial scaling could not happen up because of the cost factor involved. Then case 2, as I mentioned earlier, it is again there was a kit called as a GlowTB PCR kit. So this was designed using CRISPR-Cas12a, and it was a portable point-of-care testing device for TB, and the results were out within 1.5 hours. Now, how could the CRISPR technology be leveraged for point-of-care testing in India? If researchers or if students were looking forward to work in the area of diagnostics, I would highly recommend to focus and remain focused on point-of-care testing, whether it is CRISPR or whether it is RT-PCR or whether it is CLIA-based assays or whether it is any kind of assay, but try to develop assays which are point-of-care. The advantage of point-of-care is it can be used to near the patient or in field. While developing these assays, I would suggest the researchers or the students or prospective startup companies to always think in terms of field deployment. When I say field deployment, typical lab conditions might not be required in a field environment, but then the assay should perform in that kind of environment. That is what is the key factors regulators look at. So CRISPR for potential key diseases, CRISPR plus the RPA detects in 40 minutes, then field deployable in resource limited areas. Then infectious or vector-borne is Cas9 guided RNAs for dengue serotypes distinguishes from Zika. Because why I'm saying distinguishes from Zika because there is an overlap of gene sequences when it comes to co-infections. So that is why Cas9 guided RNAs clearly distinguish both the differences between Zika and the dengue serotypes. Now the advantage is we don't require a thermal cycler. We just need a visual readout based on the fluorescence or color. We are giving gradations here. Now, strategies to advance POCT, as I told you, the integrate with isothermal amplification, which is RPA or LAMP, and you should try to use battery-powered devices. In India, why I'm doing address India needs, because in India we have very difficult and tough field conditions. So rural TB screening or monsoon vector surveillance during the monsoons, like for diseases like malaria, chikungunya, and dengue, that becomes difficult. But if you can develop POCT tests, point of care tests using these technologies, that will be really commercially scalable also. So Tata and ICMR have a joint program for startups and newcomers or technologies who are willing to develop these kind of technologies. If anybody is interested, we can connect and facilitate that commercial scaling of these products, including the regulatory support. Then again, continuing, there are some challenges while doing adopting the CRISPR technology. This is off-target effects. What happens is when you are actually trying to develop the complementary RNA sequence for the intended DNA using the guided RNA, due to the technology, due to the inherent nature of CRISPR-Cas9, what happens is it leads to mutations or off-target effects. So there will be intermittent disturbances in the sequence that we are trying to develop. So this is one of the weaknesses of CRISPR technology. Off-target effects always keep coming in the sequence that we are trying to develop or design and develop. For that, what we have to do is we have to support it with validated data. And the most important thing is all these off-target effects are now being controlled by the newly developed in silico tools. But as I said earlier, these in silico tools on system, they work fine, they look fine. And when you actually do the docking studies, they actually fit into each other. But when you go to the wet lab and do the experiments, we need to do a lot of validations. Only then we come to know how much is the gene sequence, or how many base pairs need to be added, or how much kind of volume of reagents and samples need to be taken in place. Now, the POCT impact is it reduces diagnostic delay for about 2.6 million TB cases per year in India. If we succeed in having a POCT diagnostic test device for TB, this is what we can help and do. Now, what are the skill set requirements or core technical skills required? As I said earlier, molecular biology, how to design a guided RNA, how to handle the enzyme, not only Cas enzyme, handling enzymes in a laboratory, I need not tell you. There are a lot of cold chain requirement will be there, even while you are handling sensitive enzymes like Cas or any other laboratory grade enzymes. One thing I would recommend you is always keep a portable ice pack on your workbench, so that the enzyme, the cryo tube or the cryo vial in which your enzyme is having, should be always kept in the ice pack on the table. And this is one technique where no teaching is done in colleges or universities. You take the enzyme from the fridge, you put it and you put it back in the fridge. But actually, when you come to the industry, what you observe is it is taken from the freezer and the same temperature is maintained even on the workbench using cool packs or ice blocks. And as soon as you pipe it out the required volume of enzyme, again that enzyme has that vial or the PCR tube has to go back into the same cool pack, because the enzymes, as we all know, are temperature sensitive, and any kind of variation in temperature will ultimately have impact on the design or the testing outcome. Now, diagnostic developments, as I told earlier, isothermal amplification is what the principle is. There are reporter assays, and you also need a good quality management system, which is 13485 based. Then you need to have biostatisticians or automated statistical software which will do the data analysis for you in terms of sensitivity, specificity, and other KPIs for validation. Having said that, CRISPR technology is still not viable fully. Why? Because, as I said, inherent technical limitations. One of the problem is offset targeting is the main problem that happens when you actually do the wet lab sequencing and then you try to design and develop your sequence. The offset effects are there. Then ethical concerns are there. That is, you need to take prior approvals as to why this new sequence and for what kind of implications it will have in your study. All those things need to be taken, approvals need to be taken at the university level or the clinic level or the hospital level, wherever you're intending to use this. There are diagnostic challenges because of the offset target limits. There are diagnostic challenges. The intended sensitivity and specificity is not acquired. So that is why I said the validation of your sequences is very important when you actually finally land up doing the experiment. Then therapeutic challenges, again, because of the instability in designing the designing the CRISPR sequence using poorly developed sequences, especially the guide RNA. That is the problem, where with the guide RNA, the PAM complex along with Cas9, these are the three important complexes. If these complexes don't complement each other, in addition to if they don't fit together into the DNA, they don't sit properly into the DNA complex that we intending to cut and then duplicate or mutate. There the problem lies. That is the problem. That is the area the challenge lies. And most of the wet lab people who are doing these experiments actually know what I'm trying to say. That is the important, sensitive and troubleshooting area that most of the researchers or the newcomers don't take care, and they repeatedly keep doing these mistakes until they realize that they're spending a lot of money in buying these valuable, highly valuable enzymes and sequences. So these challenges are significant. They are currently being addressed through development of, for example, base editing and prime editing. Again, this which we all know. These are the important components of any CRISPR sequence study, which do not rely on double-stranded DSBs, which I mentioned earlier, DSBs, and may offer safer therapeutic options. So what I'm trying to say here is, if you know the correct option of base editing and prime editing, then you need not rely much on the DSBs. What is the importance here? Then inherent technical limitations. As I said, the photosynthesis, photoreceptors, they are the motifs, anti-motif, adjacent motifs. Their requirement is a prior requirement to your sequence that is required. So CRISPR requires a specific short sequence known as protospacer adjacent motif near the target DNA. So whichever target DNA we are trying to target, so there should be a PAM near the target sequence. Only then the complex, which I was trying to tell earlier, only then the complex sits, the CRISPR-Cas9 complex sits exactly near the target DNA along with the PAM, and then the cutting actually happens in a very nice manner. If there is any kind of concentration change in, or the sequence change in the, or even inefficient sequence, amounts of nucleotides in the reagent that you're using, the cutting may not be effective. So this requirements limits the number of available target sites in the genome. Now, longevity of effect, ensuring that the edit remains durable over the patient's lifetime and does not result in delayed unforeseen consequences, is a major safety concern. So once it is done, how long will this be effective in the, for example, we have done a CRISPR insertion, and then the new sequence, if it doesn't remain active for the intended purpose, particularly in therapeutic areas, then there is that could even lead to safety concerns. That is, the sequence could enter into any kind of mutations and create major problems. So proper validation of your sequence before actually using at the final end is more important here. Ethical concerns, as I mentioned earlier, the potential for unintended inheritable changes in human genome raises significant ethical, social, and legal issues, leading many jurisdictions to ban or strictly regulate germline editing. Germline editing is your basic ingredients of your basic DNA or RNA sequences. If you are trying to fiddle with the main components of the gene or the cell, that is not accepted unless until it is beneficial publicly or any kind of agricultural purposes. Then the diagnostic challenges, again, contamination and sensitivity. When combined with nucleic acid amplification methods needed to detect low levels of pathogen, CRISPR-based diagnostics like Sherlock or Detector face high risks of carryover contamination. In some cases, insufficient sensitivity. Now, why do you think this carryover contamination is taking place? Because of initially what I mentioned is the complex of Cas9, the PAM, and the guide RNA not sitting in the correct intended sequence in the DNA sequence, and then cutting it at the wrong places or insufficient places. That is the place where that is the problem. Where while you are doing this in silico studies, it looks very nice. It looks very neat, and you actually do the docking and you get the sequence correctly. But when you go and do it in the wet lab, you need a lot of validation to be done. Technical complexity and automation. While early CRISPR diagnostics showed potential integrating sample preparation, amplification, and readout into single step, user-friendly and fully automated sample-to-answer point-of-care device is technically challenging. As I mentioned earlier, preparation, amplification, any kind of these things will be is again dependent on the initial complex that we try to prepare, and if any kind of concentration of reagents or sequence that we are not able to handle carefully, it is more failure is more bound to happen. See, in RT-PCR, we still have a chance of recovery or reco, but that's not the case in case of CRISPR, because here directly we are dealing with the gene base pairs. Then the stability of components. Maintaining the stability of Cas enzymes and guide RNA components in field settings without relying on cold chain transport is a significant obstacle of commercialization. The point to be noted here is cold chain support, which I was mentioning earlier. Here, reagents like your Cas is very temperature sensitive, and if they are not handled very carefully, ultimately that is going to give you problems. Then therapeutic challenges, insufficient delivery mechanisms. So you have a CRISPR-Cas9 protein and guide RNA to correct the cells and tissues within the human body, in where it remains the biggest hurdle. Vectors like adeno-associated viruses have small capacity, while non-viral methods like lipid nanoparticles, they struggle to reach specific tissues outside the liver. So what happens is when you have the most trusted in most of the researchers, adeno-associated viruses are used as a carrier vehicles, and they're successful also. But when it comes to delivering CRISPR technologies, this is having a problem when it comes to non-viral methods, particularly, that is LNPs, and often struggle to reach the tissues. That is because the binding inheritancy or the capacity of CRISPR molecules to these LNPs is not still understood clearly, as well as it is not effective. Off-target effects, which I had been mentioning repeatedly. This is again, while you are developing the sequence, there are mutations or there are new kind of unintended sequences that are coming in the way, and then these kind of they trigger unwanted mutations in the body, then or in the cells. Then immunogenicity, again, this is a new problem. This isn't a new problem. It is well known to all of us, such as, for example, in staph or strepto, in which the human immune system is recognized as foreign, leading to immune responses that render the therapy ineffective or cause adverse effects. So when you are extracting Cas9 protein from bacteria, such as this thing, so what they do is the human body thinks, okay, it is a new thing which we need to deal with, and the attack happens, the immune response happens from the human body, and then any kind of therapy to counter this is failing. So low repair efficiency, or which I had mentioned earlier while doing the HDR methodology for CRISPR, correcting gene defects. CRISPR relies on homology-directed repair, which is generally low efficiency in most adult cells. Commercial and regulatory hurdles, this also you might be aware. It is the CRISPR reagents and consumables are very costly, and they require customized patient-specific workflows. So this results in extremely high cost-specific costs, sometimes exceeding even 2 million per dose, which creates barriers to patient access and reimbursement. So in India, we don't have reimbursement, but in countries where reimbursement is a part of healthcare, or insurance is a part of healthcare, even they are finding it difficult to implement the commercially available therapeutic CRISPR-based technologies. Regulatory frameworks, which I told you earlier, these were designed for smaller molecules and not for complex gene therapies. So what happened is, when we bring every new researcher or every new sequence comes up for the regulatory approval, the regulator is not prepared. Okay, now something new has come up. So small molecule, in the case of other molecules, like even I would say biopharma molecules also, after some time the therapy and the line of indication is understood, and then. But in case of CRISPR therapies, every time there is a new sequence and a new rampantly developed a CRISPR sequence, and as long as CRISPR is concerned, the regulator is not confident in giving approval because of the offset effects from the gene on the gene that is coming to happen, and the regulator asks lots and lots of validation data, particularly with regard to your gene validation. There are also intellectual property disputes, which are ongoing legal battles. There are a lot of ongoing legal battles with regard to CRISPR technologies. But as I mentioned earlier, if any of you are there in the bioinformatics segment, this is a place where you can actually use your skills and expertise. This is you need to maximizing target specificity. You need to know how to minimize off-target effects and optimizing the delivery and activity of Cas-guided RNA or the ribonucleoprotein complex. So unlike general genomics, these skills must prioritize the precise interaction between guide RNA and nucleic acids or DNA and RNA within complex genomic environment. As I mentioned earlier, the designing of guide RNA is the most complex thing. If you fail here, then there is a problem. So the designing of guide RNA and then validating of that guide RNA by repeatedly doing laboratory experiments for the nucleic acid that you intend to cut, that is where your validation, once you have done the validation there, only then move towards the other thing. Advanced guide RNA design and optimization, as I said earlier. Off-target analysis, this is proficiency in tools like Cas, Finder, CRISPR. So these are tools were already available, and they are available in the online tools. You need to use these tools for those who have aware, they know how to do it. Those who are not, I shall be sharing this presentation to the organizers. You can take it from there and probably do your hands-on experience on these tools. So how to handle off-target detection and safety assessment. This also, you have to have a lot of bioinformatics tools. This requires a lot of repeated dry lab activities. You need to sit in front of your system and repeatedly to use these sequences, and then you will come to know how it actually functions. For data analysis, these are the tools that are available. After post-experiment editing, evaluation, diagnostics, and therapy. This is for diagnostic and therapy. For example, NGS data, sequencing, sequence analysis, or repair outcome prediction. There are tools where you can do your analytics. Specialized computational skills, automation and pipeline development. You need to know briefly. You should know the language, which is Python or Bash, and structural bioinformatics tools. This is a summary of the table for what task, which tool you need to use. This I have tried to summarize in a table and present it here. Those who are aware of this, it is easy for them to use, but those who are not aware, as I said earlier, you need to sit and practice these tools. But students who can actually practice these tools and learn these tools repeatedly will only succeed in, will find better opportunities, job opportunities in the industry. Now, I would like to say about the global clinical advancements in CRISPR technology. What is happening, since I said that in my topic, I will briefly touch upon the clinical advancements that is happening across the globe. So it has transitioned, the technology has transitioned from laboratory research to transformative, approved and experimental therapies for previously untreatable genetic disorders, diseases, and cancers. The most significant milestone today is the Cas9 XFL in late 2023, the first CRISPR-based medicine for sickle cell disease and transfusion-dependent thalassemia. Very deadly diseases, but CRISPR has brought relief to these patients. So these are some of the approved therapies and genetic diseases. I leave the presentation with the organizers. You can take it from there. In cancer immunotherapy, these are the CRISPR solutions. And in therapeutic areas, there are emerging solutions that are coming up. And these are the advancements in delivery and precision. So what kind of tools you can further sharpen, and what kind of tools you can use to study, and the areas you need to study, these are mentioned here. However, these are challenges and future outlook. Again, this is one thing, off-target effects are the main thing. So repeatedly, I would suggest who's are interested in using CRISPR, you use dry lab activities more. And once you get validation from the dry lab activities, when I say validation from dry lab activities, try the same sequence against established DNA sequences, but try the new CRISPR sequence in various lengths and various formats. When I say formats, 2 to 4 base pairs, or 2 to 8 base pairs, or 4 to 6 base pairs. Give a complementary, appropriate sequence to the DNA sequence that you are intending to work against. Thank you. So here I would like to conclude here. Any questions that we can take at the end of the second presentation? Thank you.
Thank you, sir, for the side presentation. The precision and potential of CRISPR-based diagnostics are truly transformative. And as you said, we can take the questionnaire at now also, or if you will to take it at the end of the second session, we can do it then.
Okay, if anybody is having any doubts, you can ask. Otherwise, I would like to, in fact, I would like to move on because I have something else to attend to. If any questions, I would be happy to take. Otherwise, the second presenter can go on.
Can I ask a question?
Kindly tell your name and from where you are, please.
I am Juhi Maulik Parikh from Ahmedabad itself, currently pursuing BSc Microbiology in 6th semester in Silver Oak University. I have a question in molecular biology and genetic engineering, that is CRISPR gene review paper if we have written based on whatever we have studied. Can we share with you for collaboration?
Yeah, definitely. You can coordinate through the organizers of this event, and they will put it to me, and we can.
Yes, but for that, can we do a part-time internship or part-time job in that sense, unpaid or a paid stipend?
See, these things I don't know. That is what I'm telling you. The organizers are the right people to do this. I am just the resource person who is actually coordinating various kinds of research and other kind of publications, help to PhD students or postdoc students and assistants. This is my role in the industry. And about internships and other projects, you can ask the organizers. They will be in a better position to tell you. However, if it comes to me through the organizers, I will be more than happy to help you in whatever capacity I can.
So we should mail to whom should we share?
Miss Juhi, can you we can speak it on the over the call and we can take it ahead.
Madhaviji, I would request you to leave your email ID in the chat box.
Sure, sir.
I think that is a better option. You leave your email ID in the chat box so that I will also note it down. So if there is any kind of questions or students or participants would like to address, they can write to this email ID, and from there we can coordinate it.
Okay, sir. Sure.
Excuse me, sir.
Yeah.
Yeah, I am Dani from University of Sri Jayawardhanapura, Sri Lanka. Okay. I would like to ask a question regarding cancer therapy using CRISPR technology. Is it possible to use for cancer treatment as it involves multiple genetic mutations?
Invoking or inducing multiple genetic mutations in silico studies is definitely possible, but the challenge here is, as I said earlier in my presentation also, you need to actually validate these things in wet lab. And in fact, to validate, before going to validating in wet lab or using costly consumables and reagents, I would request you to kindly do the in silico studies using the tools that I've mentioned. And this presentation will be available with the organizers. You can access it through them. The tools I have mentioned in the presentation there, use those tools, validate your technologies, because what happens is, if you have multiple gene sequences, for example, I'm telling you, if you have multiple gene sequences, and if you're trying to target it using those sequences, you are trying to use one particular sequence against those many targets. You try today, it will give you a different score. When you try after some time, it will give you a different score. So what happens is, every time you pick up a database sequence from a database, that sequence you need to fix as to, if I am asking for example, if I am asking for cervical cancer gene, and in that cervical cancer also there are various types of cervical cancers, and in that cancer, which particular protein sequence or how many types of protein sequences are you targeting, against which CRISPR gene or target that you are designing. These things can be fiddled around on the in silico space, but this you cannot do on the in vitro space because of the cost factor of the reagents and consumables. So what you do is, you do the necessary trial and error method using public databases and available tools for CRISPR-Cas9.
Okay, sir. Thank you.
Hi, sir. This is Anis Siji from CRISPR Bits, Bangalore.
Yeah.
So you just mentioned in one of your slides that one of the reasons for CRISPR technology not being viable is the cold transport. So what I would like to add to that is that we are a CRISPR company working on molecular diagnostics and editing. So we have lyophilized all our reagents. So they are room temperature stable. You don't need a cold storage for transport. I'm just mentioning this, but my question is something else. So you also mentioned that there's an ICMR Tata model that helps in regulatory support. How does this work?
See, what they did is, if you actually go to ICMR website, there is a project space in which it's funded by Tata. When you write to the ICMR people for your project, you need to present the pressing cause of the gene or the design that you are invoking. And then if it is publicly relevant only, then they are encouraging. If it is for a research purpose only, they are not encouraging. They are only encouraging things which is going to have a public impact. So if you have any such ideas, you can reach out to me. Maybe I can have a basic screening of what you want to do.
Yeah.
Then if I find it feasible, because I'm also on the recent ANRF panel, the Diagnostics and Tools panel. I'm an expert member for the Task Force Committee. And the first meeting for the Task Force Committee is coming up on 5th, 6th and 7th of March, and in Delhi. So, there are about 1,200 projects, and I'm told there are a few CRISPR-based projects also there. So, I would say, first of all, route it through some experts like us. If we find it that it is of public importance, then probably we can, tell you, recommend you, as well as push resources in ICMR telling that why don't you try this.
Yeah, sure, sir. Thank you. We'll contact you soon.
Right.
Sir, we have two questions in the chat box from Dr. Ravi. How scalable is CRISPR for large-scale industrial or agricultural use? And another question is, what are the biggest technical bottlenecks still limiting CRISPR efficiency?
I think, Ravi, both of them I have answered. See, how scalable is CRISPR? CRISPR is definitely scalable, but the major problem is if you are intended, the design of your guide RNA and the CRISPR RNA, the complex along with the necessary associated enzyme, whether it is Cas9 or 12 or 13 or the subsequent polymorphs of these proteins, if they are not designed well, then scalability is a problem, because in the initial validation phase itself, if you can't, then there is a problem. That is why I told repeatedly, use these databases which are available in public domain, and the best part is they are free. These tools are free. You should have only good thing. The only challenge is you cannot use your routine your laptops or the routine using computer systems for data processing, because these sequences are large in number. When you process large amount of sequences, they generate large amount of data, and the processing time and the energy, I don't think your normal laptops will be able to handle. Because when you actually put through the screening process, once you have the sequence in hand, and when you try to validate, you will have to screen through a lot of public databases, and that needs resources like GPU, not CPU. So GPU resources, computing resources is one challenge that most of the colleges or researchers face. So this is one problem. Then you're asking technical bottlenecks limiting CRISPR is, again, I would like to come back to the same point. Designing your initial complex, like for example, in RT-PCR, your master mix is the whole game. So here the complex of the Cas9, the guide RNA, and the PAM, these are this complex going and fitting exactly into the PAM sequence that is available on the gene. That is the challenge. So try to do it in silico as much as possible, and when you feel that repeatedly you are consistently getting a score of e-score of or a score of 98% and above, only then try wet lab. If you are not getting 98% or above, don't try wet lab. Even 95, 96 I would not recommend, because definitely you will fail in wet lab. 98% and above, then probably you can think of your first wet lab experiment. Thank you.
Thank you, sir, for addressing those questions, and thank you for our audience for your engaging participation. And now we move on to our next session. It is my pleasure to introduce Professor Lanka Undigora, Professor in the Department of Biosystems Technology, Faculty of Technology at the University of Sri Jayawardhanapura, Sri Lanka. Professor Undigora's expertise spans microbiology and biotechnology. Her research includes functional probiotic foods, microbial biofertilizers for sustainable agriculture, bioremediation using pollutant-degrading microbes, and the biodiversity of yeast in Sri Lankan wine fermentation. Her work significantly contributes to food security, environmental sustainability, and industrial biotechnology. Ma'am, we are delighted to have you with us. Please take it ahead.
Thank you very much, Madhavi. So I'm Professor Lanka from University of Sri Jayawardhanapura. I would like to share my screen. Can you see my screen?
Yes, ma'am. It is visible.
Thank you. So actually here when you observe something, that means when you track on your gut, sometimes you might think your digestive tract is only for digestions. So today I would like to give you some idea about beyond the digestion, how your gut help you to being healthy. So for that, before moving on to the lecture, I would like to give you a brief idea. What is this? If you see your gut like this, you can see, oh, this is only for digestion. So it seems like you have a pouch without money. It's useless. So if you think this gut is with microorganisms, not few trillions of microorganisms, so that is the part of this which improve your health. So that is where this gut microbiome concept is introduced into this world by scientists. The reason for that is today's world we are talking about different type of hormones. We call it as happy hormones. So sometimes you might think the serotonin and dopamine, those are the hormones. Most of the time the 95% of serotonin is handled by gut. So that's why scientists say it as, yes, of course, this brain, our second brain is this gut. So now you have something in your mind, yes, rather the digestion, gut should do something more. So today's topic is to find out what is this specialty from gut beyond the digestion. So we'll see. So that is where every time today's world, people are very busy with the busy schedules. We are telling like we have dementia, depressions, anxiety. Those are common terms we are getting towards us. So in with these terms, every time all these terms are linked with our gut microbiome. So that is where I would like to give you idea about what is this important role in this gut microbiome towards your health. We'll see what is this gut microbiome. So when it comes to gut microbiome, actually it's a diverse unit with several type of microorganism which can ability to influence your digestion, immunity, metabolism, and also for the host health. So in this gastrointestinal tract with microorganisms, they can influence on all these activities, but without microorganisms they cannot influence on these activities. That's why I previously mentioned, yes, of course, if you take the only gut, it's like a pouch without money. because the pouch needs money to you to handle everything. So because of that, the gut microbiome need microorganisms. In here, most of the time, when you see the gut microbiome diversity, it's with trillions of microorganisms, belongs to thousands of bacterial species. So there are so many genes, more than 3 million genes, which handle every particular item in the body, in its brain to toe. So that's why every time when we consider, you're familiar with some terms about this, some species belongs to this gut microbiome. If I say like this, lactobacillus plantarum, bifidobacterium, privotella, so these are the common terms every time you are hearing. So this is about microbiome. So in this microbiome, so now you might think how this microbiome develop inside of your body. So it's coming from your birth. Scientific evidence reveal, if you are the your birth coming from the vaginal delivery, your microbiome is quite good. But when it compared to that, the scientific evidence proved when there was a cesarean delivery, definitely your microbiome totally different to the vaginal delivered the base microbiome. So not only that, when it comes to after the birth, the next the time period you're passing with infant's age. So in this infant's age, every time if you feed your children with milk, every time your children are having with a good microbiome. Good microbiome means diversified microbiomes. In next couple of slides I will explain you how I can say it as good or bad. So when it comes to sometimes some infants get formulas instead of natural milk consumptions, solid food and so on. There are some deviations of the microbiome coming from the milk consumption and other formula consumptions. I am telling this because since birth you are developing your microbiome inside of your body. So that will enhance your intelligence, your activities, your body health, everything are determined by all this microbiome diversity. So diversified microbiome means it gives you good neurotransmitters, good metabolites which improve your health. So that means you can think more, you can be happy, you can you are so active. So all these things basically based on gut microbiome. So now you can think, okay, beyond the digestion there are actually some sort of more things are happening around this gut microbiome. So after this birth and the infant time period, the toddlers move on to some environmental conditions. They faced different harsh environment, sometimes good environment, basically based on environmental condition. Now little by little your microbiome will be changed. So that is the development, evolutionary era of your personal gut microbiome. Why I'm telling it as a personal gut microbiome, it is actually unique thing, because it's like your fingerprint. So your gut microbiome is unique to you. So that's why you get unique characteristics to you on. So there are common microorganisms in the microbiome, but there are interconnections, symbiotic associations develop all the activities and metabolic activities and so on. So that is where every time when it comes to gut microbiome development, your birth is a really critical parameter, and other thing is the effective point is infants, the milk consumption and formula consumption. So because of all these things, it modulate your microbiome well defined ways. It should be a diversified, otherwise it will give you bad effect as well. So in the next couple of slides I will explain you Okay, this is where we should go when it comes to gut microbiome. It's with several type of good bacteria and as well as bad bacteria. Why I'm telling this, when it comes to your body, I told you more than 1,000 species are there. When it comes to more than 1,000 species every time, it's with the mixture, bad bacteria and good bacteria. The good point is there is a good balance in good bacteria and bad bacteria. When there's an imbalance, we call it as a dysbiosis. When it comes to the dysbiosis stage, you're not in healthy condition. You are getting different type of disease and infections when there is imbalance. Imbalance means every time the bad bacterial population, the colonized bacterial population inside of your gut are getting increased compared to the good bacteria. When it comes to that level, every time you are not in healthy levels, every time you are getting infected with some sort of disease and infections, and also some genetic disorders can be in a way to your body. So because of all these things, every time you have to maintain a balance microbiome. But the thing is, our food habits really impact on your balance of your microbiome. Why I'm telling this, sometimes your food habit is with huge amount of sugar, sometimes huge amount of salt. Sometimes once you get a small symptom of fever or a cold and cough, just by looking at that, you take huge amount of antibiotics into your body, and as a result of that, what you do is you destroy good microorganisms in your body. As a result of that, bad microbial population getting increased. As a result of that, you are getting different type of health issues after that. As an example, I will tell you this, sometimes the most of the times for some disease and infections, we are getting antibiotics. After getting antibiotics, there are more evidence found by scientists and reveal all these facts and figures. After getting continuous antibiotic doses into your body, it will destroy bad bacterial communities in there. As a result of that, sometimes you get depression conditions, sometimes you are having high anxiety. Why? Your serotonin levels and dopamine levels drastically getting reduced because of this imbalance. As a result of that, the no happy hormones mean every time you're worrying, you're having some sort of depressions, you're having with OCD. There are a lot of issues you are having with because of this imbalanced condition of the microorganisms. So that's why you have to balance it. Once you balance, you are getting positive effects, but once you break the balance, we call it as dysbiosis lead you to different type of negative effects. So there's some sort of gastrointestinal disorders and metabolic syndromes. That nowadays we are having different metabolic syndromes because of the diabetes, obesity, and the very common thing, kidney diseases. All these things can be happen because of this imbalance of your microbiome. Not only that, but I have to tell you, if you have a strong balanced microbiome every time, you can see your immune system is perfectly pitched to protect you from the whatever the pathogen attacks into your body. And also your brain development is very perfectly pitched, and also the pathogen invasion issues you cannot have. If it's so, you have to every time look into balance this microbiome. That's why sometimes when you want to do something in a good way, every time you are telling it as, oh my God, I want to go with my gut feeling. We said, my gut feeling says I have to do this. So what's this gut feeling? Yes, of course, gut feeling is really important. That's why we are telling it as the gut is the second brain of us. So the brain, other than brain, the gut is where which handle every emotion, every characteristics in your body. So sometimes the in the serotonin and dopamine activity, every time you can see the serotonin secretion is totally handled by gut. Every time the serotonin activity, the secretion is totally handled by gut. So when it comes to that, you can get, if you want to get a mood stabilizer, so you are in a happy mood, very enthusiastic, no laziness, the art, every time you want to do something good, so that is because of the serotonin. So that is why the gut microbiome, so there are different type of microorganisms responsible for this. There are bifidobacterium varieties, lactobacillus varieties, and we call it as firmicutors. Most of the firmicutors, lactobacillus plantarum, lactobacillus rhamnosus, like all these microorganisms handle all these things and generate metabolites. Those are some sort of neurotransmitters which are handled by this vagus nerve, brain to gut axis is handled by this vagus nerve. So every time this vagus nerve balance your body and give you protection, no anxiety, you're in good mood every time. When you're having an imbalance in your gut microbiome, that means bad microorganisms are high, and these bad microorganisms the are every time affecting your good microbial community, that because of they can generate these type of neurotransmitters, metabolites, stimulators to give you strength to your brain. So as a result of that, what happen is every time you are getting some worries, you're worrying in your life, no happiness far away from you, so depressed. Sometimes you might think, I can't do all these things, anxiety is with you, you don't like to go the being public, so every time you want to isolate into a one place and thinking more and more, bipolar disorders, everything happening because of this. That's why people are saying, if you want gut feeling, you have to maintain your gut microbiome in a well balanced manner. So unless you can't balance all these facts and figures in a very well, so other than that, actually but when it comes to this dopamine the secretions, it's basically from the brain, not from gut. But the thing is, every hormone, every enzyme in the body needs some stimulation. So that stimulation base, all the chemical compounds are generated by the around gut. So in the gut, every time this microbial stimulation, we call it as short chain fatty acids. When we have more and more short chain fatty acids, it maintains the liveness of your cells. When you have more live cells, it avoid the methylation. Actually, when there is the inhibition into methylations, mean there is slow aging process. You can be young every time. So in this scenario, you can see the balance in this microbiome. It will help you to improve all these the activities in your body, and you're perfectly pitch into this natural environment, because you are a part of natural environment. So you have to do something to the natural environment. So from the natural environment, you are getting microbiome. So that diversified microbiome protect you as every time. When it comes to dysbiosis, that means you don't have a balanced microbiome. There are so many pathogens, that means bad microorganisms are there. As a result of that, you don't have good microorganism in enough way into your gut. So there are some challenges them to make serotonin and all the neurotransmitters and metabolize into your body. As a result of that, most of the time these scientific findings reveal So asthma, disease, Parkinson, depression, and all the neurodevelopment based the weaknesses are happening because of this dysbiosis. So that's why I'm telling you this brain gut axis is really important. If you want to be in health, every time you have to maintain that. But the problem is today's busy schedules, we are having several junk food, so huge amount of proteins, you don't have enough amount of fibers into your body. You don't have fibers mean there are no food to microbiome. So as a result of that, they cannot survive. The good microbiomes getting distracted as a result of that. Bad pathogens colonize inside of your body, and their number of population getting enhanced. As so you can't get healthy life to your body. Every time your food pattern is matter of fact to this all these balances. So the most of the time with the busy schedules, you are there for lunch, for dinner, for breakfast, you are getting a small snack, the or fast food or a junk food. So as a result of that, huge amount of fat plus proteins, but no fiber based polysaccharides, the complex carbohydrate into body. Without that, these we cannot expect all these microbial communities to be developed. So that's as a result of that, every time you are getting different type of weaknesses into body. Not the other than this gut brain axis, the important point is your immune system actually. So when it comes to immune system, if you have a very balanced microbiota, every time you can expect a good immune system. You know this microbiome prohibit invasion of the pathogens into your body. So in using this strategy, every time when they cannot control the invading point of these pathogens, and at that last point they give the command to your T cells and immune system cells to, okay, now I cannot control this point, you have to be ready to control this point. So then immune system boost up and doing all these things. Simply I can get an example. If there is two children, infants, the toddlers, the one is with the environment, he is touching soil, the dirty soil and everything he is touching, and he is living with the environment. Okay, but the other students is other toddler is you can see he's every time the parents try to keep him inside of the house, not to touch soil or dirty soil or whatever the thing, not let to go out and do his own works and every time. So hygiene conditions are high 100%. But that the toddler is not like that. He is going to the outside and playing everywhere and touching every environmental the stuffs. And so that means that little by little the resistance to the pathogens and develop some sort the develop inside of the microbiome, and it improve the immune system very well. So that's why every time when you get to know about the environment, and when you live around the environment, most of the time it's not a perfectly hygiene condition. So it's with harsh conditions, it's with different type of pathogens, it's with several type of the unfavorable conditions. It lead to improve your immune system. That's not because of improve the immune system. Totally what happen is you are getting a diversified microbiome inside of your gut, because once you have the food, once you inhale, every time you are getting different the immune system developments because of some sort of diversified microorganisms in your outdoor environment. So that is where every time functional tuning, immune system functional based protection is you are getting because of this microbiome improvements. Now you can see when you look at the world, the region to region, there are different type of food habits. The Asians, they have different food habits. When it comes to Western world, they have different food habits. Sometimes you can see when it comes to Western world, most of the time they prefer to have fat and protein enriched food items. So when you analyze the scientific evidence proved there are a broad range of variations of these diversified microbiomes, the region to region. The reason for that is mainly our food habits, the secondly environmental factors. Why it matters the food habits, you can see in this first picture. Every time when you have more and more protein and fat-based food items, it enhances the bacteroids diversity. That means bacteroids are the microorganisms which prefer to eat fat and protein based food substrates. They prefer on that. So, but when you see the South Asian region, sometimes most of the time, especially in the ancient time period, you can see this Protella bacterial community. The reason for that is they prefer on fibers. When you have Protella based community, you are lucky, very lucky to get a diversified immune system based protection, gut and brain axis based protection. The reason is they have high skill to generate neurotransmitters, metabolites which stimulate immune system and give you the protection, and which enhance the serotonin, the secretions, which gives you the strength to live. So all these are happening because of the diversified metabolites which can be generated by this Protella, the microorganisms. But when it comes to Western pattern, but they prefer protein plus fat, it leads for the bacteroids collections. When it comes to bacteroids collections, it's really depend on the fat and protein, not on the fiber-based food substrates. And in that end, what happens is every time they have some narrow ability to generate some sort of chemical compounds which enhance the immune system, which enhance the brain gut axis and all the things. So this is how. But when it comes to African countries, again you can see diversified microorganisms which basically based on the marine and environmental collection that also stimulate some sort of immune system protected chemical compound generations. So as a result of that, you can now get the idea. So in dispersed globe, basically based on food patterns and busyness in their life, all these things lead to some sort of destruction in the microbiome, and also some regions, they because of their good food habits, it improve your microbiome and give you healthy conditions. So this is the global trend. When it comes to this global trend, most of the time, because of high sugar consumptions, high salt consumption, and also high doses of antibiotic consumptions lead to the dysbiosis. What is this dysbiosis? Imbalance of your microbiome. It leads to the latest finding, the evidence like stroke risk, brain stroke risk, and depression risk. And every time you are getting all these things because of this dysbiosis. So if it's so, you have to find out some sort of solutions. Why? To improve your gut microbiome. Now you are having a deteriorated gut microbiome. The next thing is you have to follow up your gut microbiome. Now you might think, how I can improve my gut microbiome? Yes, of course, you can improve your gut microbiome. When it comes to this improving, I would like to divide that into two categories. The first one is untargeted improvement, and also we can go for a targeted improvements as well. So the doctor's recommendation is to do exercise more. So when you do exercises, you are getting enough amount of oxygen, your metabolism is very good, so that means you are giving good substrate, nutritional substrate to development of microbiome. So when you get a good fiber enriched foods to your body, other than the protein and the fat, instead of that, if you can eat more and more grains and other fiber based food items, that will improve your microbiome in a well manner. So other thing is the latest world invention is this fecal microbiota transplantation. So actually nowadays we are talking about that, we call it as a FMT therapy, but still I would like to say this is not from this modern world invention. Actually, most of the past people practice this concept and improve their body. If as an example I would say, in ancient China and other countries, most of the time when they have a sick person, what they do is they detected some people who is with a good health condition. They believe they are the one who are good to donor the stools, fecal matters to the person who are in the bed with sickness. So that is the concept used previously in the ancient era, since the very ancient time period. But nowadays, latest invention is we are moving towards this FMT technologies. We are using fecal matters. We are getting fecal matters from the very healthy person. So then we transplant them to the people with the different type of genetic disorders and other disease, infections and all the things. If you refer the papers, you can see how far this is very successful strategy we follow to cure the diseases in this world. Many more evidence we obtain from here. So other than that, actually the targeted concept every time in this targeted concept is with bioengineered commensals. You know, the nowadays we are practicing this phage therapy and CRISPR Cas9 strategies. I will explain all these things. So what is first I would like to move this phage therapy. You know, when there is a viral attack, so that they develop bacteriophages to control the body, and in the end of the day they lyse the cell and destroy the cell to get into a stabilized level. So this is we call it as they follow a lytic and lysogenic cycles to control all these things. So same concept we are using here to control the bad microorganisms in our microbiome in this world. What you can do is using this phage therapy, you are giving the bacterial phages which can control which can ligate the genes into the bacterial cells. We identify them as those are the pathogenic bacteria. So those are the pathogenic bacteria which you need to destroy. So using this bacteriophage concept, what you can do is you can add these phages, when they develop the phages, these bacterial cells getting destructed as a result of lysis in the end of the day. So you can destroy the pathogens in your microbiome in a selective way. So other than that, actually the CRISPR Cas9, the dysbiosis strategies like this, most of the time you can see we can do this in two ways. So one way is the destruction of the pathogenic bacteria inside of the microbiome. Other thing is the improvement of the some sort of bacteria which gives good facts into your body. So two ways you can do it here. The gene knockout therapies they are. So as a result of that, every time what you can do is, now you have a good background about this CRISPR-Cas9 the concept. So that because of here we use to improve gut microbiome, gut brain axis, so which we have microorganism, as I told you, which can stimulate the secrete the improve the serotonin secretions and the dopamine secretions. So if you can establish them in the microbiome using the CRISPR-Cas9 therapy, so you can improve them using that gene improvements. And other thing is, if there's a pathogens, you can knock down their genes and you can destroy them very well. So in these two manners, you can go control some sort of Alzheimer's, Parkinson diseases, depressions, and some cancer therapies nowadays we are looking for. Using most of the time, microbiome is the responsible genes which provide this Cas9 enzymes. So that's why in the Cas9 there is a specificity issue. That's where today's world introduced this Cas9 the enzyme system with this CRISPR system to control the microbiome improvements, based improvement you can do. So when it comes to this, the next one is I told you untargeted improvements, how we can do for the disposes issues in the microbiome. So in this concept, the first thing you can see is here, fecal microbiota transplantation. So in this world there is a concept, we call it as a yellow soup concept. So we are looking for donors which is with good health, so you can get stools from their fecal matters and that transplant into the people who are getting different type of disease resolutions. So in this concept, most of the time the ancient people, they practice actually the sometimes they don't, in the ancient era they didn't have enough amount of medicine. At that point, what they did was actually they use this yellow soup concept, and they got some fecal matters from a healthy person and transferred that to the patients who are having the diseases, like different type of disease effects. That's how it happens. So when it comes to all these things, now you might think, okay, so some prefer FMT, some doesn't like to go for FMT. That's how the patients are like that. So when it comes to that concept, today's world, the scientists introduce some improved concept as well. So this is the probiotic, and then the prebiotic, and then symbiotic, and the postbiotic concepts are there, which can improve our microbiome. What we do in here is, we don't have enough balanced microorganisms inside of our microbiome. As a result of that, what you can do is, one thing is, you can take the microorganism from the outside and get them into inside of the body and let them to stabilize there. That is the concept behind this probiotic. In this world, there are different type of dairy and non-dairy probiotic products. What you can do is, there are so many pharmaceutical products basically based on this probiotic concept. What you can do is take them, and once you take them into your body, they will establish in your body to improve your microbiome. So in that aspect as well, you have to find out whether how these microorganisms can survive in the microbiome, because microbiome in your body is coming from your birth. So once you get the foreign things into your body, foreign microorganism into your body, you better check whether they can survive in there or not. There are many ways to check that as well. So after doing that, you can get your own probiotic formulas nowadays. That much of improvement we got into the place, so that because of there are so many improvements in this probiotic concept. So as far as you know the probiotic, the second concept what you can do is you have imbalance in your microbiome. What you can do is you have few amount of good microorganism and high amount of bad microorganism. What you can do is provide as much as suitable food for good microorganisms to colonize well in there. That's the second thing you can do. That concept is called prebiotic. What you can do is by getting so many fibers, complex carbohydrate and all the things, what you can do is provide suitable substrate to be establish all these good microorganisms in your body. So that is what you do is you are getting food from outside into the body which are suitable for the microorganisms inside of your body. So that is called prebiotic. So nowadays with the busy schedule, you can't take food at the suitable for you. You used to get so many junk foods, the high sugar components and everything. So no way of probiotics. So that's why scientists thought of symbiotic as well. What they did was they externally in vitro condition in vitro conditions they developed probiotic microorganisms, and they grown all these microorganisms in a suitable fiber enriched substrates. Those two things, that mean microorganism plus foods are there. So what you can do is as a capsule or the food item or a nutraceutical, you can get that compact item with the food plus microorganism, and after getting that into the body, they will settle out and improve your good microbial community, and that will help you to keep in healthy conditions. So that concept is called symbiotic. Far beyond that, nowadays world moving towards the postbiotic. As I told you, there are different type of chemical compounds which generate by these microorganisms in your body. That means your microbiome is the community which generate all the required receptors, neurotransmitters, metabolites, which enhance your protein degradation and everything. What happens is they secrete bioactive compounds. What you can do is, if you know these bioactive compounds, whether it is short chain fatty acid or whatever the thing, what you can do is you can generate them in vitro and take them into your body. So that means you grow the microorganisms, that means microbiome related microorganisms in the lab, and let them to secrete chemical compounds. Then you extract them well into the pure and suitable formulations, and then you can get back them into your body. This is called the concept postbiotics. So how we can handle all these things in, if you want to improve your gut microbiome, you have to get them externally in a way of probiotic. If not, you can give the suitable substrate. It's called prebiotic. So both together, symbiotic or else chemical compounds that you want, this is called postbiotic. So in a whatever the way, what you are going to do is improve your microbiome. So I told you, as you wish, you can't get microorganism. The reason is when you take a yogurt, it's with lactobacillus, bulgaricus, streptococcus. Sometimes those are not probiotic organisms. If your product is termed it as a probiotic only, those are probiotic. All the lactic acid bacteria are not probiotic organism. The reason for that is, if you define whatever the organism under the probiotic characteristics, how you can do is they should have is acid tolerance, bile salt tolerance, and what they have to resist to digestive enzyme. Why? Once they have all these things, all the foods with the probiotics, the microorganism into body, there are a lot of enzymatic conditions in your digestive tract. Whatever, once they active, they can destroy the cell structures of microorganisms as well. So it's useless. So that's why we need to get microorganisms who can resist against this digestive enzyme. Sometimes once you have so many foods into your body, it's with huge acid amounts, bile salt amount. So if they cannot tolerate that level, definitely they cannot survive. So in that term, you cannot get their advantage in terms of improvement of your body. Why? Once they face to that acidic condition, they will get destroyed. It's useless. So those type of microorganisms are not, you cannot term them as probiotics. What you can do is they should be with the characteristics of acid and bile salt tolerance. So other than that, there are effective adhesive properties. There should be. The reason is, for a day you are taking huge amount of water. Once you get microorganisms and simultaneously when you with the tablet, when you get water, what happens? It's washing off. It can be washing off. So if they don't have the adhesive properties, easily they can wash out. So that's why when you term a probiotic, definitely they should have it effective adhesive properties towards the gut lining. So unless, you cannot name them as a probiotic. So every time they need to colonize well. Within a short time period, they should colonize. Otherwise, they cannot survive. So multiplication of the microorganisms are really important when they settle out in whatever the environment into their climax population. So that's where, so other thing is it should be non-pathogenic. Once you get a probiotic with, in terms of pathogenic, mean you are improving the bad microbial community in your balanced microbiome. That will be happen. So as a result of that, once you get all these things and balance, so you can see most of the time through the probiotic concept, we are getting bifidobacterium, lactobacillus, and some sort of in terms of lactobacillus acidophilus, plantarum, rhamnosus, there are so many organisms which improve your skin, which improve your immunity, nutritional status, and oxidative stress, and all the things. It improve your body in a well manner. So latest world is with gerobiotics, not with the probiotic, not with the postbiotic, not with the symbiotic. Latest term is gerobiotic. Actually, this is really important. There are microorganisms who can make many more short chain fatty acids which stop the methylation activities and which inhibit the aging activities in our body. So if you want to live long time without aging, so this gerobiotic is the concept nowadays world introduced to not to get the aging concept. It inhibit the aging activities in your body. So the most of the time these findings basically, in our population, there are people who are getting age quickly, and also some people are really tolerable into some sort of life cell degradation scenario. So using all these, comparing all these microbiome, they introduce some sort of special formulas into this gerobiotic nutraceutical concept, which can enhance the short chain fatty acid stimulations, the production stimulations, and they are trying to control the aging activities in your cells. When it comes to that, the modulate of histone modifications and all the DNA methylation activities, they stop and manipulate well inside of the cell. So the today's world move on to the personalized probiotic using these dispenser machines. Once you go there, you can add your saliva or stools into there and give you a profile. So then this is my microbiome. So then you can think, okay, if there is a high pathogenic microorganism, that means the bad microorganism, how you have to improve your profile. So then they are giving you a suitable nutraceutical unique to you. because once you go to the market and take a probiotic yogurt, it's a common scenario. You are getting a common microbiome into your body. But when it comes to here, this is totally personalized, unique to your microbiome. They are the first the identify the microbiome inside of your body, and thereafter what they do is they personalize these probiotic concepts and give you to improve your microbiome. That's how this personalized probiotic concept, all these are commercialized products nowadays you can focus on. So now I think you might think about beyond the digestion, there should be something more in your gut brain microbiome. The gut brain microbiome is where which control your healthiness and improve your genetical disorders. If you have diabetes, that may be the reason for that, may be sometimes imbalance dysbiosis of your microbiome. If you have obesity, that's totally because of these imbalances. And also sometimes if you have some sort of sometimes because of these imbalances, it gives the brain strokes and some dementia status, depressions, anxiety, and the Alzheimer's, Parkinson's, like different type of issues can be happen in your body. So that is about the gut microbiome. I think you got something from this session. Thank you very much.
Thank you, ma'am, for that fascinating exploration of the gut microbe and its role in systemic health. We will now open the floor for questions. Attendees may ask their questions directly. And in the meantime, we will take a screenshot of our webinar. Please raise your hand or indicate in the chat if you'd like to speak.
Hello, good evening, madam. I'm Dilan from University of Sri Jayawardenepura. I'm having a little bit of a question. Like, as you mentioned in the principles of FDR, or like taking microbiomes into your gut system. So the system basically revolves around taking microbiomes into the gut system from external. So what I'm asking is, given those conditions that our bodies goes through, like whether it could be a disease or in a specific condition, our body also changes with us now. So what my question is, even though we get microbiomes from the external, would they be able to survive in the same conditions that the body is still in? Like, would they be able to survive in the same conditions that the body is in? The condition which gave away the microbiome to be destroyed in the first place.
Yes, you meant the total recovering point. So the thing is, when you take probiotics into your body, that means the microorganisms from the outside into body, it's quite the there are some different conditions in there compared to the external environment. Definitely it's a totally different condition, but what they do is they are little by little adapting into that particular environment. So when that's why, so the first time when you take the probiotic, little bit of recovering point you are getting, but second time, third time, and continuous consumptions and the involvement of these microorganisms inside you can improve your microbiome community inside of your body. But the thing is, total recovering as in the previous months means sometimes you are taking special microorganisms. That means Lactobacillus plantarum, Lactobacillus acidophilus, like few couple of microorganisms we are taking. But I finally explain you the personalized probiotic concept. In there, what we do is we identify the who are the microorganisms previously were, and then compared to that, we are trying to introduce the total microbial community. In that case, it's really easy them to get into there and survive. But when you take common probiotics into your body, it takes time to survive because it's a totally different environment. But slowly what they do is try to recovering by settling around all these environments. That's how it happened.
Thank you, ma'am. Thank you for addressing the question. And I request all to switch on your cameras so that we can take a screenshot of our session and we may post it on the social handles. Request all the to switch on their cameras. Thank you, ma'am. Thank you so much once again. And thank you to everyone who participated today. That concludes today's Synexis Global Research webinar. If you have any more questions, you may write to us at contact@synexis.org. We truly appreciate your time and engagement. We look forward to welcome you to our upcoming sessions. Have a wonderful day. Thank you so much, ma'am, once again. Thank you.
Welcome. Thank you very much for organizing this type of very important session, you all, and your effort and everything. This is really good things to the society. We can convey the messages which people need. This is what we have to do. Thank you very much for that.
Thank you, ma'am. If a participant have any questions, we will direct to your mail. Okay. Signing off for today. Thank you, everyone.